Hello, everyone. Thank you for attending. Today, I'm really honored and excited to be able to share my research with you on better understanding brain pathology and cerebral malaria so that we can come up with better ways to treat it. So malaria continues to be a significant cause of sickness and death. And cerebral malaria is particularly devastating because even when patients are treated with drugs that effectively kill the parasite that causes cerebral malaria, a lot of patients could die. And for those that survive, they can develop neurological sequelae. And so we really need new approaches to better treat cerebral malaria. And host-targeted therapeutics are an attractive class of drugs to consider as adjunctive therapy for cerebral malaria. And in the context of this disease, they could help to dampen hyperinflammation and mitigate host pathology. So where would it make sense to target these drugs? One side of significant pathology, which I think would make sense to target, is the endothelium. So under normal circumstances, the endothelial layer in our blood vessels behaves as it should, which means it's helping to inhibit coagulation and allow the free flow of blood through vessels. It's also maintaining barrier integrity. But in the context of cerebral malaria, there's significant accumulation of parasite-infected erythrocytes, which accumulates in small blood vessels. There's coagulopathy that occurs, and then permeability of the endothelium. And in addition to parasites, there's evidence of excessive thrombin signaling, which contributes to pathology. And then some patients will go on to develop severe brain swelling, which is strongly associated with death from cerebral malaria. And we don't really understand how brain swelling develops, but breakdown of the blood-brain barrier has been implicated in this process. And so again, this really puts endothelial dysfunction at the center of brain pathology and cerebral malaria. And my research really seeks to understand how disruption of the blood-brain barrier occurs and how this can be repaired. So from studies on regulation of the endothelial barrier, it's known that there are three interrelated processes that work together to maintain barrier integrity. And integral to these processes are protein kinases. But we have a limited understanding of how cells transduce signals to effect changes of the barrier. And then thinking again about host-targeted therapeutics, can kinase inhibitors restore barrier function? So to answer these questions, I have used a kinase-focused systems biology approach known as kinase regression. And this work was done in collaboration with Alexis Koshansky's lab at Seattle Children's, and I worked with Ling. So how does kinase regression work? First, you perform a small-scale kinase inhibitor screen to see how these kinase inhibitors alter the phenotype that you're interested in studying. So in this case, changes in the barrier regulation. And these kinase inhibitors have been selected because together they inhibit many different kinases in the human kinome. And from this published data sets, we know what the target profile of each of these kinase inhibitors is. So in the set of 300 human kinases, we know all the different kinases that each inhibitor is targeting. And so together, these two sets of data are used to generate a model which then makes predictions on informative kinases, so kinases that are important in regulating the cellular phenotype that's being investigated. And so for the first step in kinase regression, the kinase inhibitor screen, what I did was use a simple but useful model of the blood-brain barrier. So I grew primary human brain endothelial cells in a monolayer, and then I monitored changes in barrier integrity using a device. And for this inhibitor screen, I induced barrier disruption using thrombin. So I mentioned thrombin earlier. It's an important clotting factor, but it's also a pro-inflammatory stimulus, which is important in cerebral malaria pathogenesis, but then also other inflammatory conditions. And in vitro, what thrombin does is induce a rapid increase in loss of barrier integrity. So that's indicated by the negative cell index. You reach this point of maximum disruption, and then there's restoration back to baseline of the barrier. And at the point of maximum disruption, by microscopy, we can see that there's retraction of cells, the gaps that form, which are indicative of barrier permeability. And so for the screen, I induced barrier disruption and then followed up shortly with the panel of 28 kinase inhibitors. And I was pleasantly surprised to see how well some kinase inhibitors worked to blunt the barrier disruptive effects of thrombin and promote recovery to baseline. And here I want to draw your attention to this purple kinase inhibitor, bosutinib. It's an FDA-approved drug. It's approved for the treatment of leukemia. But while I was working on this project, there was a paper published that showed that it attenuates vascular leakage in a mouse model of acute lung injury. And so it could potentially be beneficial in the context of cerebral malaria. So to compare the activity of the 28 kinase inhibitors, I measured the area under the curve for these from these barrier function assays. And these are the results for the 28 kinase inhibitors, where on one end of the spectrum, I can see kinase inhibitors that are barrier protective. And I highlighted a few on the left here. And then on the other end of the spectrum, there are kinase inhibitors that actually worsen the barrier disruptive effects of thrombin. And this differential modulation, differential activity is helpful for the model to predict informative kinases. So the data from these screens were fed into the machine learning model, which predicted that several kinases, 50 kinases, were important in modulating brain endothelial barrier integrity. And so what I did was select a subset of these predicted kinases to confirm a role in barrier regulation. And here I'm showing just a few examples of the kinases that I validated. So what I did was knock down expression of each of these kinases separately, and then perform barrier function assays to see how each knockdown line fared in the context of thrombin. And I found that knockdown of some kinases worsened the thrombin barrier disruptive effect, whereas knockdown of CSK was barrier protective. Now, the knockdown phenotypes of CSK and MATK cells was curious to me because both of these kinases are negative regulators of Src family kinases. But then both by barrier function assays and then also microscopy, I can see that they have opposing phenotypes. And I think this really warrants further study. But at this point, seeing how well kinase regression worked to identify barrier protective kinase inhibitors and barrier regulatory kinases in the context of thrombin, I was really eager to use the same approach to better understand the barrier disruptive effect of parasite-infected erythrocytes. So in the literature, it's been reported that parasite-infected erythrocytes directly induce barrier disruption, and I'm able to reproduce that effect in the barrier function assay. So you can see a dose-dependent increase and loss of barrier integrity. And then I can also see this by microscopy. But to me, the really interesting question is, what is driving parasite-mediated barrier disruptive signaling? And so applying the kinase regression approach, I performed the kinase inhibitor screen, and it was really heartening to me to see that some of the same kinase inhibitors, which I found to be protective in the case of thrombin, also attenuate barrier disruption induced by parasite-infected erythrocytes, even though thrombin and parasite-infected erythrocytes are very distinct barrier disruptive stimuli. And across the 28 kinase inhibitors, I could see a strong correlation in inhibitor activity, comparing thrombin and parasite-infected erythrocytes. So at this point, I've really set the stage to be able to investigate kinases that are predicted to be important to modulate brain endothelial barrier integrity. And I can conclude that kinase regression works well to investigate the regulation of barrier integrity. And then genetic depletion or chemical inhibition of host kinases can strengthen or weaken endothelial barrier integrity. And then importantly, kinase inhibitors can protect against both parasite-infected erythrocytes and thrombin-mediated barrier disruption. And bosutinib is of particular interest, and there's a promising lead for further investigation for therapy for cerebral malaria. But my work is not done. There are many outstanding questions that I'm interested in answering, which would help with targeting host kinases for therapeutic benefits in cerebral malaria. So first, how is kinase activity coordinated in time and space to result in either barrier disruption on the one hand or barrier repair in the context of parasite-infected erythrocytes, but then also host inflammatory stimuli? And this work would focus not just on the kinases, but then the substrates that they act on. And then I'm also very interested in investigating how endothelial cells sense parasite products and how this is linked to barrier disruption. And then finally, in longer term studies, I'd like to investigate the activities of bosutinib and PKR inhibitor to see how they result in barrier protection. And with that, I'd like to thank everybody that was involved in this work, including my postdoc advisor, Joe, who was willing to venture into new territory with me, and then Molly and Corey, as well as Alexis for introducing us to kinase regression, and Link, who's been really great to work with. And thank you all for your time and attention, and I'm happy to connect with anyone via email. Thank you.